Hello friends, welcome to this video on the pathogenesis of the glomerular injury. Mechanism of the glomerular injury. The main mechanism is the immune mechanism, and this is the mechanism which underlies most of the primary and the secondary glomerular disorders. Now the immune mechanism, it has been studied under antibody mediated, cell mediated immune injury and activation of the alternative complement pathway. Out of this, the most important one is antibody mediated injury. So in the antibody mediated injury, the main factor is the formation of the immune complexes, that is antigen antibody complexes. So these complexes, they get deposited in the glomerulus. So once they get deposited in the glomerulus, they initiate inflammatory reaction. And when there is an inflammatory reaction, we have the infiltration of the different inflammatory cells. And these inflammatory cells which come there, they produce the mediators, chemical mediators, which will cause damage to the tissue, and that leads to the glomerular injury. So we will be discussing about the types of the immune complexes and the locations of the immune complexes, how they produce the inflammatory reaction, and what are the inflammatory cells which are involved in producing the glomerular injury, and also about the mediators which are involved in the glomerular injury. So when we see this antibody mediated injury, as I told you, it is because of the antigen antibody complexes which are formed. Now in this we have two types: in situ immune complex deposition and circulating immune complex deposition. In situ immune complex deposition means here the antigens are present within the glomerulus itself. So antibodies are formed and they come and form an antigen antibody complex within the glomerulus. So antigens are present within the glomerulus. Whereas the circulating immune complex deposition means here the antigens and the antibody complex are formed somewhere else in the body. They are present in the circulation and then they get trapped in the glomerulus. So that is a difference. And when we talk about the in situ immune complex deposition, again we have two types in this: fixed intrinsic tissue antigens and planted antigens. Fixed intrinsic tissue antigens means here the antigens are the components of the glomerulus itself. The particles which are present in the glomerulus itself, they act as an antigens. Whereas the planted antigens means these antigens are present somewhere else and they get trapped in the glomerulus, they get planted in the glomerulus. Then we have the antibodies formed against these antigens. So this we call it as a planted antigens. Now in the fixed intrinsic tissue antigens, as I told you, antigens are present within the glomerulus. So we have two important substances which act as antigens. One is NC1 domain of type 4 collagen, which acts as an antigen. Now this is the normal component of the glomerular basement membrane, which acts as an antigen. And another one is phospholipase A2 receptor antigen. Some other antigens are also there, but in the fixed intrinsic tissue antigens, these two are very important. Whereas in the planted antigens, we have again two types: endogenous and exogenous. Now endogenous means these are formed within the body. So we have tumor antigens, or the DNA substances or nuclear material, which are the substances which get trapped in the glomerulus and acts as an antigens. And exogenous antigens are the antigens which are from the outside, like we have the microbial products. So these products also act as an antigens. They get trapped in the glomerulus and then we have the antibodies formed against these trapped antigens. So in the planted antigens, we have endogenous and exogenous. So first we will study about fixed intrinsic tissue antigens. So as I told you, we have two types in this, that is phospholipase A2 receptor antigen and NC1 domain of type 4 collagen. So this phospholipase A2 receptor antigen, this is present on the membrane of the visceral epithelial cells or the podocytes. This is specifically M-type phospholipase A2 receptor. This is present on the epithelial membrane of the podocytes or surface epithelial cells. This one acts as an antigen. Now here what happens, we have the antibodies which are formed against these antigens, These antibodies cross the basement membrane and they form the antigen-antibody complexes on the cell membrane. They are attached to the cell membrane initially. Then once we have the formation of this antigen-antibody complexes, there is the activation of the complement system. Once the complement system is activated, we have the factors like complement factor C3 and the C4. So these antigen-antibody complexes along with the components of the complement, they form subepithelial deposits. Actually, the immune complexes, after combining with the components of the complement, they shed off from the cell surface and they form subepithelial deposits of the immune complexes. So the subepithelial deposits of immune complexes, they contain antigen-antibody complex along with components of the complement. We have C3 and C4 also here. Now this is a light microscopy picture. So in the light microscopy, we cannot make out whether it's subepithelial or subendothelial, but we can just make out that there is some thickening of the basement membrane. So that's why the term is used as a membranous nephropathy for this condition, because we see the thickening of the glomerular basement membrane. In the electron microscopy, you can see the deposits here. This is the basement membrane. On the outer surface of the basement membrane, we have the deposits. This is the capillary lumen and this is the visceral epithelial cell. Outside it is outside the basement membrane. And when we see in the immunofluorescence, we can see the granular deposits. If you see here, you are seeing some granular deposits of the antibodies. This is not a linear deposit, but it is granular because we have subepithelial deposits. In between, we have the food processes of the podocytes. So here and there, we have the deposits, which gives this granular appearance instead of the linear appearance on the immunofluorescent microscopy. So that was about phospholipase A2 receptor antigen, which is present on the cell membrane of the surface epithelial cell. So the next one is on the fixed intrinsic tissue antigens. Next one is NC1 domain of the type 4 collagen. This also acts as an antigen. So this is a component of the collagen. This collagen is the important component of the basement membrane. If you will see my the previous video, which is on the normal structure, there I have already told that glomerular basement membrane is made up of, specifically the main structure is made up of the type 4 collagen. On that we have attachment of the glycoproteins. So this NC1 domain of the type 4 collagen, it is uniformly distributed along the basement membrane. So the antibodies which are formed, they are also uniformly distributed along the basement membrane. They don't form any any deposits here. Now these intrinsic fixed antigens, they cannot be mobilized to form the discrete complexes. Instead, we see the diffuse deposition of the antibodies. So we have antigen antibody complexes which are uniformly distributed in diffuse manner along the basement membrane. So this is an electron microscopy. This entire thing is a basement membrane. You are seeing this deposits here. And if you see the immunofluorescence, we have a linear pattern of deposition of the antibodies. So here it's not granular, it is linear because antibodies are also diffusely present along the basement membrane. Now another important thing is this NC1 domain, that is non-collagenous domain of alpha-3 chain of type 4 collagen, which acts as an antigen. It's present in the blood vessels. So these antibodies which are formed against this NC1 domain of type 4 collagen, even they cross-react with the basement membrane material of the blood vessels which are present in the lungs. So we have glomerulonephritis which are affecting the kidney, and we can also have the involvement of the lungs. So when we have this cross reaction and involvement of the lungs, this condition we call it as a Goodpasture syndrome. And this condition, the glomerulonephritis, by this one it accounts only for the fewer than 5% of the cases of the human glomerulonephritis. So if you have involvement of the kidney along with the lung, where the antigen is NC1 domain of type 4 collagen, then the condition we call it as a Goodpasture syndrome. This was about the fixed intrinsic tissue antigens. We have finished two things, that is phospholipase A2 receptor antigen and NC1 domain of type 4 collagen. The next one was in the in situ formation of immune complexes, that is planted antigens. So these antigens, they are present in the circulation. So when they are present in the circulation, when the filtration occurs, they get deposited in the basement membrane, they get deposited in the mesangium, or some of them may get deposited in the subepithelium also. This depends upon the charge, the physicochemical properties, and the size of the particles. So different things are there which determine where they are deposited. So in this, the antigens which get deposited, we have some cationic molecules, DNA, nucleosomes, the nuclear proteins, bacterial products, large aggregated proteins like aggregated immunoglobulins, or immune complexes themselves. They have a free reactive site which can combine with the antigens or the antibodies. So even the immune complex themselves, they can act as an antigens. So these antigens in the circulation, they can deposit in any site. So the site at which they are depositing, it depends upon the charge, their physicochemical properties again. So these antibodies which are formed against these antigens, slowly they form the antigen antibody complexes in different locations. So we can have the subepithelial antigen antibody complexes, or we can have in the basement membrane, or we can have in the mesangium also. Now when they are present in the subepithelial membrane, when we see in the immunofluorescence, it has the same pattern as we have seen in the phospholipase A2 receptor antigen. So we cannot make out, or we cannot differentiate this one by using the immunofluorescence staining. Now that was about the fixed intrinsic tissue antigens and planted antigens, that is in situ immune complex. The next one was about the circulating immune complex deposition. Now in this, the immune complexes are present within the circulation. So antigen antibody complexes are formed somewhere else. They are present in the circulation, but as they reach the glomeruli, they are trapped in the glomeruli. Now this one, the trapping occurs either because of their physicochemical properties or the hemodynamic factors which are very peculiar for the glomerulus. So these immune complexes, that is circulating immune complexes, typically they are deposited either subendothelially or they can be deposited in the basement membrane. So this is about the circulating immune complexes. Now in this again we the antigens which form the circulating immune complexes, we have endogenous antigens and exogenous antigens. So endogenous antigens which form the antigen antibody complexes are, as we see in the systemic lupus erythematosus and in IgA nephropathy, whereas the exogenous antigens are the microbial antigens. So we can have some microbial products, bacterial products, or the viral products to which the antibodies are formed, and we have antigen antibody complexes formed in the circulation. So they will be circulating in the blood flow, and once this blood reaches the glomerulus, they get trapped in the glomerulus. Similarly, we have antigens of Treponema pallidum, Plasmodium, and several other viruses. So out of these mechanisms, like in situ immune complex and circulating immune complex mechanism, the most common cause for the glomerulonephritis is the planted antigens, to which we have the antibody formation and there is a subsequent immune complexes. That is the most common formation. That's the most common cause for the glomerulonephritis. Now that was about the immune complexes, how they are formed. Now what are the factors which affect the glomerular localization? So glomerular localization of the immune complexes and the antigens or antibody depends upon the size and the charge. So size of the circulating immune complexes is, if they are very large, they may not reach the glomeruli itself, but they can be removed by the mononuclear phagocyte system. And if they are smaller immune complexes, they reach the kidney and they are nephrogenic. Whereas when we see the charge of the reactants, if they are highly cationic, they form subepithelial deposits. If they are highly anionic, they form subendothelial deposits. And if they are neutral, they form the mesangial deposits. This is because highly cationic antigens, they tend to cross the glomerular basement membrane, which has anionic components. So because of this, the cationic antigens, they can cross this basement membrane and form subepithelial deposits. Whereas highly anionic macromolecules, they have a repulsion from the glomerular basement membrane because they also have the anions. So they are excluded from the glomerular basement membrane and they are trapped subendothelially, or they are not at all nephrogenic also. They may not form any deposits also because the glomerular basement membrane doesn't allow these particles to settle here. So highly anionic may form subendothelial, highly cationic may form subepithelial, highly anionic may form subendothelial deposits. Whereas the neutral antigen antibody complexes without any charges, they cross off this basement membrane also and they get deposited in the mesenchyme. Now that was the charge and the size which determines the location where it has to be deposited. So we have seen how the immune complexes are formed and how they are getting deposited. Now what happens once these immune complexes are deposited? Now once they are deposited, they elicit inflammatory reaction. And why this inflammatory reaction occurs is whenever there is an antigen antibody complex formation, there is activation of the complement. There is a binding of the complement and there are the factors of the complement which are released. These factors, they produce inflammatory reaction. Along with that, there is engagement of these Fc receptors on the leukocytes and glomerular mesangial or the other cells. So these cells also, they release the mediators. So glomerular lesions, they exhibit leukocyte infiltration. Then we can have the proliferation of mesangial cells and the endothelial cells. So now we'll be saying how the location of the immune complexes affects the inflammatory reaction. Now, if they are subendothelial deposits, now they are more accessible for the leukocytes which are present in the circulation. So these subendothelial immune complexes, they can have interaction with the leukocytes and they can produce the activation of the leukocytes and they cause inflammatory reaction. Even the mesangial deposits, they can also produce the inflammatory reaction. They are also accessible for the macrophages which have entered into the mesenchyme. Only the subepithelial deposits, they are not accessible to the inflammatory cells because we have a basement membrane which acts as a barrier. So this basement membrane doesn't allow these inflammatory cells to come in contact with the subepithelial deposits. So usually the subepithelial deposits, they doesn't produce any inflammatory reaction. So we have a non-inflammatory pathology in case of subepithelial deposits, whereas the subendothelial deposits and deposits of mesangial matrix, they produce the inflammatory reaction. Now, if the antigen is for a short time, it's a short-lived or limited, then the inflammatory cells which are coming here, the neutrophils, monocytes, macrophages, whatever the inflammatory cells are coming, they engulf these immune complexes and remove them. And once these immune complexes are removed, inflammatory reaction subsides. But if the immune complex deposition is for prolonged period, as we see in the SLE or the viral hepatitis, then what happens? The inflammatory reaction becomes a continuous process. We have a continuous deposition of the immune complexes which will be producing the inflammatory response, and you have a continuous release of the mediators and the proteases by these inflammatory cells. So repeated injury occurs due to the continuous inflammatory process. So that leads to chronic membranous or membranoproliferative type of the glomerulonephritis. So if the antigen is short-lived, damage doesn't occur because it is removed by the inflammatory cells. But if the deposition is for a longer time, then definitely it produces more damage, producing chronic membranous or membranoproliferative type of the glomerulonephritis. Now, as we have seen, we had the mechanism of the damage is by the antibody mediated or cell mediated, or you have activation of alternative complement pathway. Cell-mediated immunity, they thought that this is also one of the mechanism for glomerulonephritis because there was an evidence for the presence of activated macrophages and the sensitized T cells which cause the glomerular injury. So they thought that this is also one of the process. And in some conditions like dense deposit disease and C3 glomerulopathies, they have seen that there is an activation of the complement pathway. So these are the mechanisms of immune mechanism. So when we have this, all the depositions, then what are the cells which are involved? Means what are the inflammatory cells which come there causing the damage. So the inflammatory cells or the cells which cause damage are the neutrophils, macrophages and lymphocytes, platelets and the mesangial cells. Now why the neutrophils should come there is, as I told you, when we have antigen antibody complex formation, there is an activation of the complement system. So this activation of the complement pathway, it produces C5a and C5b, the end products. And C5b, it forms a complex with the C9. C5b, C9 membrane attack complex is formed, whereas C5a, it acts as a chemotactic factor for the neutrophils. Along with that, there will be also Fc mediated adherence, and then there will be activation of the neutrophils. So once the neutrophils come there and they get activated, they release the proteases, which will cause damage to the basement membrane. They release a reactive oxygen species, which will cause oxidative stress and cell damage. They also release the arachidonic acid metabolites, which will reduce the glomerular filtration rate. So these are the effects of the neutrophils which infiltrate there. Then we also have the infiltration of the macrophages and the T lymphocytes and the antibody-mediated immune mechanisms and cell-mediated reactions. Now these macrophages and the T lymphocytes, they release several chemical mediators which produce the inflammatory, which actually causes more inflammatory response and also causes damage to the tissues. Then the platelets. So when the endothelial cells are damaged by this immune complex deposition, what happens? The property of anticoagulant property of endothelial will change into procoagulant. So we have the aggregates, small microthrombi, and we have the platelet aggregates. So when the platelet aggregates are formed, then what happens? We have the activation of the platelets, and these platelets, they release eicosanoids, growth factors and the mediators. So growth factors, what they do is they cause the proliferation of the glomerular cells. The mesangial cells, they cause the proliferation of these cells, and the mediators of the inflammation, other mediators, they cause the vascular injury. Whereas the mesangial cells, they release a list of the factors. They release a reactive oxygen species, cytokines, chemokines, growth factors, eicosanoids, nitric oxide and endothelin. All these factors are released by the mesangial cells. Mesangial cells, when they get activated, they are enough to produce the inflammatory response even in the absence of these neutrophils, macrophages and T lymphocytes. Just the irritation of the mesangial cells will cause release of all these factors, and they themselves can initiate an inflammatory response in the absence of the inflammatory cells are involved. Then we have the mediators, which how the mediators cause the damage. So as I told you, we have the products of complement activation. So in that, the end product is C5a and C5b C9. So C5b C9 is a membrane attack complex which causes the cell lysis. C5a, it acts as a chemotactic factor for the leukocyte influx. And these products of complement, they also activate the mesangial cells. And we have seen the list of the mediators which are released by the mesangial cells and how they cause damage. Now, there are some other factors, eicosanoids, nitric oxide, angiotensin and endothelin. These factors are also produced, which produces the hemodynamic changes. Then the main cytokines which are produced are the IL-1 and TNF. These two are produced by the leukocytes which infiltrate there. Now, when these cytokines are released, they actually induce the leukocyte adhesion and a variety of other effects. They induce the leukocyte adhesion so that the more leukocytes come there, the more inflammatory cytokines are released and inflammatory response becomes more exaggerated. Then in the chemokines, the most important chemokine which is released here is monocyte chemoattractant protein 1. Now this promotes the monocyte and the lymphocyte influx. Then we have a list of the growth factors. Platelet-derived growth factors which are released by the platelets, they produce mesangial cell proliferation. Then the other growth factors which are released by the mesangial cells, TGF-beta, connective tissue growth factor and fibroblast-derived growth factor. Now these growth factors, they cause extracellular matrix deposition and hyalinization. And this vascular endothelial growth factor, it produces the proliferation of the endothelial cells and it maintains the endothelial integrity. And the coagulation system, once there is an activation of the coagulation system, the thrombin which is formed in this, it stimulates the parietal epithelial cells to proliferate and it causes the crescent formation. So these are all the these are all the soluble mediators which causes the glomerular injury. So we have seen first the immune complexes, the types of the immune complexes. Then these immune complexes, they produce inflammatory reactions. So what were the inflammatory cells coming there and what were the mediators released? Then the mediators which are released, how they cause the damage, that we have finished. Then these mediators which are released there, they cause damage to the podocytes, that is visceral epithelial cells. So visceral epithelial cells have a foot processes. If you remember, they have the foot processes. So whenever there is a release of the antibodies to the podocyte antigens or toxins or cytokines or some other viral infections, there will be damage to the visceral epithelial cells. So how they manifest is either there will be effacement of the foot processes, loss of the foot processes, or there can be vacuolization and the retraction of the foot processes, or there can be complete denudation of the foot processes from the basement membrane. So we have denudation of the visceral epithelial cell itself completely. So once the visceral epithelial cell is lost, the filtration of the proteins, actually these food processes, they are also useful for the filtration of the proteins. So once we have the loss of this podocytes, there will be the proteinuria for the patient. So loss of the podocytes we typically see in the focal segmental glomerulosclerosis and in the diabetic nephropathy. All these mechanisms we have seen. Now what histological changes we see whenever there is this immune deposition and all that we'll see here. So normally when the nephrons are getting destroyed, the GFR reduces, and once the GFR, glomerular filtration rate, reduces to 30 to 50% of the normal, then slowly the patient will progress to the end stage renal failure. Now though you remove the original stimulus also, once the patient reaches here, slowly he will go into the end stage renal failure. Now we have, as I told you, we have two major histological characteristics what we can see in the glomerular injury. First we have the focal segmental glomerulosclerosis, and then we have the tubulointerstitial fibrosis. So why this focal segmental glomerulosclerosis occurs is because of the inflammatory cells and the mediators, we have endothelial and visceral epithelial cell injuries. So we have already seen when there is a visceral epithelial cell injury, we have loss of the foot processes or denudation of the podocytes. So that leads to increased glomerular permeability of the proteins. So these proteins which infiltrate, actually they stimulate the mesangial cells. So these mesangial cells, they proliferate and they release the mediators. Not only that, the function of the mesangial cells is it also produces extracellular matrix. So when these mesangial cells are stimulated, it starts depositing the extracellular matrix, and the mediators which are released by the mesangial cells, they produce the inflammatory reaction. So initially we have a segmental glomerulosclerosis, means only a part of the glomerulus will be involved and the remaining part will undergo compensatory hypertrophy. But as the endothelial cells and the epithelial cells are damaged because of the mediators, later on the proteinuria will continue to develop and leading to the total glomerulosclerosis. The patient will develop total glomerulosclerosis, and when this develops, the patient will develop the uremia. This is the microphotograph. So you are seeing this is a glomeruli, and initially it is a part of the glomeruli which is showing the sclerosis. Later on, the entire glomeruli will undergo the glomerulosclerosis. The last is, after the glomerulosclerosis, the patient develops tubulointerstitial fibrosis. Here we have the tubular cell damage. Why the damage occurs is it can be because of the ischemia or the loss of peritubular blood supply due to the sclerosis. So we have a sclerosis of the glomeruli, so vessels are occluded. So this leads to ischemia of the peritubular blood supply because the vessels coming out of this glomerulus, they form a network around this tubules and along the loop of the Henle. So when you have ischemia, even the tubular blood supply is reduced. Either it can be because of that, or it can be because of the inflammation in the adjacent interstitium or increased proteinuria. That also causes damage to the tubular cells. So we have tubular cell damage and the activation of the tubular cells. So once we have the activation of the tubular cells, they start expressing the adhesion molecules to the inflammatory cells, and they also elaborate the pro-inflammatory cytokines, chemokines, and the growth factors. So these inflammatory cytokines and the chemokines, they attract more inflammatory cells, and the growth factors which are released there, they deposit more of the collagenous matrix. So this leads to the interstitial fibrosis and the inflammation. This is a micro photograph. If you see, these are the tubules which are lined by the cuboidal cells, and in between you are seeing the inflammatory cells, and this is all the collagenous deposition in between the tubules. So initially we have the glomerulosclerosis. From segmental it becomes total glomerulosclerosis. Later we have the involvement of the interstitium also, so we have tubulointerstitial fibrosis. So these are the histological changes which occur in the glomerular damage. Thank you, friends. I request you to please give your comments on my presentations, which will help me to further improve my classes. Thank you.